Hello, folks, and welcome back to episode 2 of As the Cell Divides. When we talk about the cell cycle, again, there are two distinct phases in the cell's life cycle. There's interphase, inter meaning between, right? So it's the period between cell divisions where cells grow, basically make proteins, do their job, and then they copy everything and get ready to divide. So interphase is the period between cell division. Again, cells will spend almost their entire life in interphase. And then there's actual cell division, which includes mitosis and cytokinesis. Now, if we want to add to the confusion, cell division is also sometimes referred to as the M phase or mitotic phase. Okay, so cell division includes mitosis, the division of the nucleus, and cytokinesis, the division of the cytoplasm. But cell division is also sometimes referred to as M phase or the mitotic phase. So good luck. Sorry, language is sometimes confusing. But anyway, cells will spend most of their time not dividing, and then they'll divide. So we'll get into what goes on during each of those parts of the cell cycle. Now, in reference to interphase, cells spend most of the time in this G1 or gap1 phase. Now, G stands for gap, but I think of it as like growth. Okay, so during G1, cells are growing, making proteins, and doing whatever in the cell they do. Okay, so cells are just basically doing their thing. Now, when signaled, cells will transition into what's called S phase, and S stands for synthesis. So what they are synthesizing during S phase is a copy of those chromosomes. Remember when we saw the two sister chromatids that make up a chromosome? Well, during G1, a chromosome is unduplicated. Okay, so we have an unduplicated chromosome in G1, and then during S phase, you duplicate that DNA, and now a chromosome is made of two sister chromatids. Okay, so you can start to imagine what types of questions they could come up with on the AP exam. After S phase, the amount of DNA in the cell should double, right? And then think about what happens during cell division or mitosis and cytokinesis. The amount of DNA should get cut in half. So, G1, you've got one chromosome. G2, it's still a chromosome, but now it's been replicated, or it's a duplicated chromosome. So you have two copies of the genetic information. So we'll get some practice with defining that type of stuff, but that's the kind of thing they can get into with that. G2, or the second gap phase, I think of it as final preparations. They're copying organelles. They're making proteins that'll be needed to help the cell divide. So the cell is basically doing like final preparations. That's what I think of it as. So G1 is when cells are just basically doing their thing. That's how they'll spend a lot of their time. S phase, they copy their DNA. And then G2, think of like final preparations, copying organelles, making proteins you need to stimulate cell division, stuff of that nature. Now, mitosis, again, it focuses on what happens with the nucleus. And there are 4 distinct phases we'll look at. Sometimes books will have like pro-metaphase and like early anaphase, late anaphase, stuff like that. For the sake of keeping it simple, we're just going to focus on PMAT, prophase, metaphase, anaphase, and telophase. Now, cytokinesis, the division of the cytoplasm, that does happen during like late anaphase and telophase. So you will see some changes that occur in the cytoplasm in addition to what's going on with the nucleus. So here's an illustration from the book. What I suggest looking at is like during interphase, you have a nuclear membrane that's fully intact, and then the DNA is relatively relaxed, so it just sort of looks cloudy in the nucleus. What I think of to myself is I don't see threads. Okay. Now, when we get into prophase, you'll notice a couple things happening. One, the nuclear membrane will begin to disintegrate. So as you move here, the book distinguishes prophase and prometaphase. You can see that the nuclear membrane dissolves. The DNA, rather than being just sort of cloudy, will actually condense down into chromosomes, and you see those threads. And then the third thing that happens is the spindle fibers are forming. And we'll talk about the value of spindle fibers later. But spindle fibers begin to form. So during prophase, again, the things I'm looking for, you see the threads because the DNA is condensing into chromosomes. The nuclear membrane is breaking apart, and the spindle fibers are forming. Now, metaphase, it's a dead giveaway, right? DNA all lines up in the middle. It does that so that the DNA or the chromosomes will split equally. You have those two sister chromatids, they're going to be separated from one another during anaphase. So meta means center. So during metaphase, the DNA all ends up in the middle so that the chromosomes can separate evenly. Ana, the prefix ana means away. So anaphase, I think, is sort of easy to identify because it looks like an action photo. So the cell's elongating, and then the chromosomes are moving. The daughter chromosomes are moving away from one another. Telophase, telo means end. So during telophase, you're basically undoing what happens during prophase. So rather than the DNA condensing into chromosomes, it's relaxing back into that cloudy chromatin. The nuclear membrane, rather than dissolving, is actually reforming. And then the spindle fibers in telophase are breaking apart rather than forming, as you see in prophase. So again, think of telophase as an undoing. All right, so now let's talk about these spindle fibers. These spindle fibers are made of proteins called microtubules. And microtubules play a really important job in anchoring the sort of reproductive structures within the cell and then separating those chromosomes. Now, there are a couple of terms here. One is the centrosome. That's this sort of cloudy area that we see here. So if you look at it under a microscope, you'll see the centrioles. They sort of look like these bundles of sticks. They're a bunch of microtubules. And then the cloudier area is around the centrosome. People still don't know exactly what the centrosome does. So, I mean, there's still mysteries to resolve here. And then what's really cool is this aster that we see. It's a radiation of microtubules out. Aster means star. And so these asters are these sort of starburst of proteins that radiate out from the centrosome, and they actually anchor to proteins in the cell membrane. So the aster serves as anchors on either end of the cell. So when the microtubules begin to shorten, it helps keep the anchors separated from one another. So the aster holds the whole complex of the centrosome in place in the cell membrane. So that's pretty cool. Okay, kinetochores. Here's a new little detail. Remember we talked about centromeres being like the narrowed wastes in cells? Well, there's a structure in there, and it's called a kinetochore. I think of a kinetochore as being like a belt or a harness. Microtubules are these long spindly proteins. They attach to the kinetochores. So it's sort of like if you've ever been rappelling or rock climbing, you wear the harness and then you have the rope attached to the harness. Well, the microtubules are like the ropes, and then the kinetochores are these proteins that act like a harness that connect to the microtubules. So here you can see an electron microscope image here of the kinetochore. And again, this will be the protein that acts like a harness, and then the microtubules attached to that. Okay, so here you see the kinetochores. They look like black jelly beans here, and then the microtubules attach. Now you'll notice that some of the microtubules connect to kinetochores, and then some of the microtubules don't. So it's pretty descriptive. The kinetochores that attach, the microtubules that attach to the kinetochores are called kinetochore microtubules. And then the microtubules that don't are called non-kinetochore microtubules. Okay. So here's why that's important. These non-kinetochore microtubules that you'll see here, they have little motor proteins. Remember little motor proteins that walk along? Well, these will have motor proteins that walk the non-kinetochore microtubules apart from one another. So when the motor, like if these are microtubules, what will happen is the little motor proteins will walk and they'll push those apart. So when you do that, it elongates the spindle complex and it stretches out the cell. Okay. Anaphase. Again, ana means away. So here's sort of the sad story where the sister chromatids separate, never to see one another again, right? So the sister chromatids separate. But before you separate them, remember that cohesin protein that stitches them together? Well, there's going to be an enzyme that cuts that up. So if the enzyme cuts up cohesin, that separates the sister chromatids, and now they're referred to as daughter chromosomes. Okay, it's still the same genetic information. They just give you slightly different names to suggest sort of what's occurring in the cell. So you have one chromosome made of two sister chromatids. Cohesin hold them together. When the cohesin dissolves and the microtubules begin pulling them away, then the sister chromatids are now referred to as daughter chromosomes, and now this becomes two distinct chromosomes. So this is one chromosome. We had two sister chromatids, and now these are two daughter chromosomes. So you can have chromosome numbers change within cells depending on the stage of mitosis. So again, imagine the fun they'll have with that on the AP exam. Okay. Let's see. So a couple things happen. You have the kinetochores, the little harnesses that connect the microtubules to the chromosomes. They will be dragged and pulled away, and so the daughter chromosomes will get pulled away from one another. At the same time, those non-kinetochore microtubules have motor proteins push them apart, so that stretches out the cell. Okay. Now, there was a clever experiment to look at the question of what happens to these daughter chromosomes. Are they dragged away from each other, or are they reeled in? Okay. And to help discern that, what they did was they added phosphorescent tags to the proteins that make up microtubules, so they glowed. Okay, so the microtubules would glow with these phosphorescent tags, and then what they did is on one side of the complex is they zapped it with a laser to get rid of the phosphorescent tags. So the microtubules were still there, but the phosphorescence was gone, so it basically stopped glowing. And then what they did was they let mitosis occur. And when the chromosomes pulled apart, they wanted to ask, okay, where does it shorten? Does it shorten to the left of the laser mark, or does it shorten to the right? Since it shortens to the right, what that suggests is that the daughter chromosomes are being dragged away from one another. And so the idea is what we see here, the motor proteins walk the daughter chromosomes down along the microtubules, and at the same time depolymerize it. They basically break it up into smaller pieces. Okay. Now, it turns out in different species, what happens, it differs. In some species, the daughter chromosomes get reeled in from the centrosome. In others, the chromosomes get dragged away from one another. And there's some evidence to suggest that both happen in other species. So it really just depends. But what is useful to be able to look at something like this and be like, oh, okay, well, since you have the laser line and you see the phosphorescence shorten, as the chromosomes get dragged away, it looks like they are being dragged away or pulled away from one another. Here's that image zoomed in. Okay. We talked about anaphase. Here's telophase. Again, telo means end. So think about what it'd be like to be DNA at the end of a long, hard day of dividing. DNA just wants to relax and unwind back into chromatin. So rather than seeing the threads of DNA, it's relaxed and you don't see the threads anymore. The nuclear membrane begins to reform, and those spindle fibers are dissolving as the motor proteins walk along. Of course, cytokinesis occurs during anaphase and telophase. Cytokinesis is about the division of the cytoplasm. Now, there's something that's absolutely fascinating about cytokinesis that occurs in animal cells. And let's talk about plant cells real quick, and then we'll get back to the animal cells because I think that's a more fun one to end on. So what plant cells will do is actually have little vesicles deposit polysaccharides, and they'll form a wall between the cytoplasm, and that will create the new animal cells. So it reminds me of, is it the Cask of Amontillado, where the guy, I don't want to give away anything if you haven't read it yet, but it just reminds me of that because you deposit these polysaccharides and then eventually it'll form a cellulose cell wall between the new cells. Now let's get back to the animal cells and the cool thing that happens there. So what happens is there are these two proteins, actin and myosin. They overlap one another, and then the myosin has these little heads, these extensions of the proteins that will row and pull on actin. So if this finger is actin and this finger is myosin, the myosin will pull on the actin, and it sort of ratchets it tighter. So this is like a belt that will ratchet and tighten and tighten and tighten and tighten until it splits the cytoplasm in two. Now, that may not seem like much, but what's remarkable is that these two proteins, actin and myosin, that are arranged in a loop and tighten like that and cinch the cell and cause it to divide, if you, rather than having them ratchet like that, you have them overlap like this and bundle a lot of them together, it's what creates our skeletal muscles. So the muscles we have evolved from these proteins that play a role in cytokinesis. So it's sort of amazing that there's this process that's been around for, again, billions of years of helping cells divide like this. And if you just take those same proteins and reorient them and make a bunch more of them, all of a sudden you can do things like create muscle that will shorten and move bones. So I think that's a really cool evolutionary connection between cell division and then sort of the structure and movement of our bodies. All right, now how our cells divide is through mitosis and cytokinesis. Bacterial cells, it's a little different because they don't have a nucleus. They undergo what's called binary fission. Bi meaning two, fissus means to split. So with binary fission, this is sort of the original form of cellular reproduction. There's no nucleus here, so you don't have all the steps that you have in mitosis. Instead, with binary fission, all you get is the DNA replicating, and then the cell membrane and the cell walls of bacteria will divide. Okay, we'll get into binary fission a little bit more next semester. The last example we'll look at in this segment is just another sort of version of cell division, and this is what happens in organisms like yeast. They'll have a nucleus. They're eukaryotic, so you see the sort of purple nucleus there, but they've got the microtubules inside of the nucleus. So they'll have their chromosomes separate, but they'll use sort of a spindle apparatus that is found within the nucleus. So it's thought that these organisms produce this new eukaryotic means of cellular division or cell division or mitosis, and then what we have is sort of evolved from that. So that's just sort of an interesting look into that. Okay, well, that's it on 2. We'll come back for part 3. How do you control all this stuff? So we'll see you soon.